So today I'm going to try and make the case that the earliest adaptively evolving systems were surface-limited metabolisms, which we call slimes, and that thinking through this, the existence of slimes provides some suggestions for some interesting sort of empirical approaches. So to get us started, let's just define the core problem. The core problem, as I see it, is that the first adaptively evolving system had to be self-propagating. Also, that system that was both self-propagating and adaptively evolvable, by definition, had to arise without the help of any prior adaptive process. It had to spring into existence spontaneously. And therefore, whatever it is has to pass what we might call the junkyard tornado test. So many of you will know that Hoyle, pointed out that we don't really expect a tornado to be able to blow through a junkyard and blow together an airplane, a 747. And by similar reasoning, even the simplest living cell is too complicated to imagine that without an adaptive process that a prebiotic soup, given some chance and time, could spontaneously form a cell. So we need something significantly simpler than a cell. Now, one alternative that has been thrown around in the field is the idea that the initial starting place might be a single molecule with the ability to replicate itself, and people usually think about RNA. So the idea here is that you have prebiotic soup, it gives rise to an RNA. That RNA can self-propagate, and the nature of self-propagation in RNAs makes it intrinsically evolvable. However, I think I won't have to convince many of you in the room that there are a lot of problems with this idea. Firstly, long RNAs are very unlikely. Secondly, we have the whole error threshold problem, that it would be even harder to get one that could self-replicate itself accurately enough. And finally, we have the whole problem that it would need to be able to make its own monomers. So I put a not equal sign there to indicate that we don't consider this a plausible hypothesis for the first evolver. So if it's not a single molecule, it has to be an ecosystem of molecules. It has to be a set of interacting molecules with the collective property that they all facilitate and achieve one another's production. This general idea, and I actually can't find the pointer. Is that it? Yes. This general idea of autocatalytic systems, or ecosystems, as I prefer to call them, is first I want to point out this is chemically agnostic. The players in these ecosystems could be RNA molecules, they could be metabolites. There's no indication or requirement that there be specific catalysis, because branch networks, such as this little loop here, because every time it goes around it makes 2 A's, the system would self-propagate, even though it doesn't have any specific catalysis. And it also doesn't require specific catalysis, but it does require, any such system would require a constant replenishing source of food and energy, because it's going to be dissipating energy in the act of self-propagating. So the challenge then that causes is that we have a set of cooperating species in this ecosystem, but which need to be maintained in physical proximity in order to cooperate and make more of one another in that neighborhood. But at the same time, they need to have access to continuing flux of food and energy. So how could they be spatially localized? So one popular view is that the initial system was a spontaneously formed protocell. And I think many people would acknowledge that given a lot of amphiphilic molecules in a aqueous solution, you can imagine the spontaneous formation of a protocell, and maybe once in a while it could trap within it a chemical ecosystem with this ability for every member of that set to cooperate to make one another. However, we would like to suggest that although this is at some point life likely went through such a stage, we don't believe this could have been the first evolver. And the reason is that while this achieves the goal of keeping the cooperators enclosed and spatially proximate to one another so that they can make one another, it also builds a barrier between them and the food and energy that they need in order to be able to propagate themselves. So it's actually making life harder by separating them from that energy and food. The other issue is that because you have now two components, they have to somehow miraculously, without any prior adaptive process, be coordinated so that they can actually grow without either the lumen contents getting diluted to non-existence or the membrane popping. So we don't think this is a plausible model, and so we want something simpler. Now, one idea just to put there that is, we would say, viable is the idea of a droplet or a coacervate. So this is similar to a protocell, except you only have one phase. So you have some sort of maybe, this is a set of amphiphilic molecules that form some sort of droplet. If the droplet is small, those molecules do still have access to the external environment where the food and energy has to come from. And so this is a perfectly viable, we think, model in principle, and may well have been the path by which life-like system evolved in some places in the solar system or the universe. But we don't think it's a good model for a precursor to cellular life, because once you have a droplet, it's very hard to see how you can go to a cell, how you can generate now a hydrophilic bubble within a hydrophobic droplet. So I'm going to set that aside and instead tell you about the one that we do think is the most plausible way to think about this, which was originally proposed by Wechtershauser in a form. So the idea here is that, again, you start with a prebiotic soup, it interacts with a mineral surface, and spontaneously you get a small set of cooperating molecules, an ecosystem nucleates on that surface and can grow and spread, can self-propagate. So just to give an example and help you visualize this, we have this little network I showed you before, a little branch network, and we're imagining that the capital letters denote species that are the cooperators, and they all stick to this membrane but are not present in the soup. And the lowercase letters are food, energy sources that are in the environment and are being constantly replenished. And so the notion is that if some rare side reaction generates one member of this set, it would stick to the surface, and it would then nucleate a system whereby these would grow out and propagate laterally to cover that surface eventually. So that's what we mean by a slime. But you might wonder, well, could a slime be evolvable in the absence of any kind of genetic encoding system? Well, in principle, there's good theory to suggest the answer is yes. Because there isn't in the totality of chemistry one autocatalytic loop system, but real chemistry has lots of loops, lots of potential autocatalytic networks, such that once you have one network, like in this case, so here we have one loop that I showed you before, this loop can feed on waste from this loop. So once that one has covered a surface, you can imagine, again, a spontaneous reaction, rare one, giving rise to some member of the red set, like that H that maybe you did or didn't see. And now these molecules can be added to this slime, to this system. So what that means is that slimes can exist in multiple metastable states that can be found by rare side reactions, the analog version of mutation. And as a result of that, these slimes will not only be self-propagating, but there would be sort of communities of these systems that would be where there could be some sort of competitive interaction and the potential for selection among them. Now, it's not the main focus of the talk, so I'm going to be very brief, but one thing this does offer is a very natural path to a cell, because once you have a slimes on a surface, they're going to be limited to their surface, but they're going to be patches of surface elsewhere in the ocean. Selection would favor any variant that could bubble off little bits of itself into the water column to spread and colonize other surfaces. Furthermore, those variants that could not only spread but use energy in the water column to divide would be favored to the point where they no longer need the mineral surface. And this was similar to what Wechtershauser himself had proposed. So we think that the starting place is prebiotic soup that interacts with kinds of mineral surfaces and spontaneously gives rise to these adaptively evolving chemical systems on surfaces, these slimes. So we think it's a promising approach. And what's nice about it is it suggests an empirical approach to the origin of life, which we think is somewhat overdue. So the experiments that we've been doing in my lab as part of this NASA-funded group, we call it the Chemical Ecosystem Selection Paradigm for Origin of Life, or cesspool, is looking for slime, very fitting, by building little microcosms of the prebiotic earth. So little prebiotic soups, minerals, and atmosphere, imagining that a slime might spontaneously emerge, and then using a serial transfer and dilute protocol to enrich for any slimes that might be present, and to detect their presence using chemical proxy traits. Now, obviously, I don't have time to tell you the results, but Lena Vincent, my co-author and student, is going to be talking on Wednesday to give you the results. But let me just say that this approach has found evidence of the spontaneous sort of accumulation of organic layers on iron pyrite grains. And at a macro scale, we see a very interesting long-term dynamic across these transfers that looks very ecological in structure, with what look like ecological booms and bust cycles. So the conclusions I would hope that you might take away is that first evolvers has to arise without any help from prior adaptation, meaning they must be simple. And we would contend that that rules out a single magic molecule, and it also, maybe more controversially, rules out a protocell as the first evolver. Slimes offer one alternative because they're evolvable and there's a natural selective path to a cell. And that these early empirical studies seem to us consistent with this basic approach. So just to end, I want to thank my entire lab, but particularly Lena, shown there, collaborators at UW-Madison. We've received funding from NASA and NSF and the University of Wisconsin. And I also want to acknowledge other members of the cesspool consortium listed here, some of whom are at this meeting. So thank you.
Thank you, David. We have time for questions.
Just speak up.
I'll repeat it. I'll repeat it.
Would you have a prior, because one problem is the problem of evolvability, as you mentioned, so you show a scheme with a second automatic network, so do you have a prior idea of what that could be?
So the question was the constraint is evolvability, and so the example I gave was a particular hypothetical ABCD network showing loops. And the question was, do we have specific ideas? No, I wouldn't say. I have some vague ideas. There's if people look in core metabolism and find hints of autocatalytic loops rather like this. The basic loop looks like reverse TCA fitsat, for example. But you need a second one and so forth. But that's sort of the approach we're taking is we give it the soup, we see what emerges, rather than trying to build a soup in order to generate a particular target autocatalytic system. So we're trying to do it in a rather more bottom-up approach, see what happens.
We probably have time for just one question. Sure.
So David, is there any particular reason you chose to work with pyrite in the experiment? There's thousands of other minerals, right?
Yeah, so our goal was to develop a protocol that could be used in a broad sweep by the whole community of different minerals, different soups. But we just decided, given Westershausen's model, to start with a soup, well, it's a Miller-Urey type soup, and we decided to, among a few we tried, and this one gave these interesting results, so we've been sticking with it for the time being. But eventually we should try many, many other minerals and soup combinations, including ones that are appropriate to other planetary, other worlds, since there's no reason to restrict them just to this Earth chemistry.